Hi everybody, welcome back. If you remember last time, we completely reviewed mitosis and all of its details, and now we are going to review meiosis and all of its details. So hopefully from the first two lectures we've given you a solid context through which all of this more detailed information can be placed within. We're just moving through the slides right now. Just as a gauge your recollection of where we just came from. All right, so meiosis. So let's start back at the locations where these processes are occurring. So as a quick review, meiosis is a special kind of cell division that occurs in germ cells. Germ cells are a special group of cells located within the female and male gonads. The female and male gonads are otherwise known as the ovaries and testes, respectively. In the ovaries, the germ cells are called primary oocytes. So here we have our ovaries here. So within the ovaries, we have our germ cells, which are called primary oocytes. In the testes, the germ cells are called primary spermatocytes. In the female, the primary oocytes will eventually give rise to a gamete commonly referred to as the egg or ovum. And then in the male, the primary spermatocytes will eventually give rise to a gamete commonly referred to as sperm. All right, so let's go over the purposes of meiosis. The purpose of meiosis is cell division of germ cells for cutting a diploid or 2N genome in half to make a haploid genome. And this is, by the way, a mistake. This should say 1N right here. All right. I'm glad if you caught that ahead of time before me, because it's the first time I'm seeing this. So cutting a 2N genome in half to make a haploid genome or 1N genome for the purpose of making gametes, aka sex cells. All right, and what are the two gametes? We just went over them. In the male, okay, we have the sperm, and then in the females we have the eggs, okay, or ova would be plural for ovum. All right, so here's a question for you. Why do gametes have a haploid set of chromosomes? Your answer is that so that when the sperm and egg combine in a process that is referred to as fertilization, right? The resulting zygote, remember we all started off as one zygote, has a diploid or 2N set of chromosomes. So if you remember, if you have in a human being, if you have a haploid set of chromosomes, you have 23 different chromosomes. You only have one of each type of chromosome. You only have one of each 22 autosomes and either an X or a Y sex chromosome. All right. But a diploid state is you have a homologous pair for each type of chromosome. You have a homologous pair for each of the 22 different types of autosomes, and then you have a homologous pair of sex chromosomes. If you're female, you have two X's, and if you're male, you have an X and a Y. All right, so that when you combine those 23 different solitary chromosomes with another set of 23 different solitary chromosomes, you now have homologous pairs for each of the 23 types of chromosomes, okay, giving you a total number of chromosomes in the diploid state of 46, all right? So now we're going to go into the specifics of these processes. So whereas mitosis is a process involving one cell division, meiosis is broken down into two sequential cell divisions. The first cell division is known as meiosis 1, and the second cell division is known as meiosis 2. We begin with an overview of meiosis 1, all right, as this sideways heading designates. Meiosis 1 begins with prophase 1. Prophase 1 shares many similarities with prophase of mitosis. All right, so now we're going to start with a germ cell. All right, which is going to undergo prophase 1 of meiosis 1. That's the context that we're working from right here. And remember, in the males, this germ cell may be a primary spermatocyte, and in a female, this germ cell may be a primary oocyte. All right, so now let's go through interphase now, and then we'll progress on to prophase from there. Okay, so as in prophase of mitosis, in prophase 1 of meiosis 1, the 46 different chromosomes with 23 pairs of homologous chromosomes begin to condense into bivalent chromosomes. So in this very, very simplified diagram, we only have one homologous pair of chromosomes. But just keep in mind, in a normal cell, this germ cell, whether it's a primary oocyte or a primary spermatocyte, as it progresses from interphase, where the chromosomes are all in the loose, unwound form called chromatin, and condense and condense and condense into the fully wound bivalent X-shaped chromosomes that we're all familiar with, you are going to result in 23 different sets or pairs of homologous chromosomes. However, in this very simplified depiction of a cell, we only have one homologous pair of chromosomes. And we're going to use the same coloring scheme that we used for mitosis, where the blue bivalent chromosome here is of paternal origin, all right? And then that the white bivalent chromosome here is of maternal origin, all right? So they're both of the same type of chromosome. They're both homologous chromosomes for this certain type of chromosome. And they are representative of only one of 23 different sets of homologous chromosomes that you would normally find in these germ cells as their chromosomes condense into bivalent chromosomes. All right. So as in prophase one of, or as in prophase, not prophase one, but as in prophase of mitosis, in prophase one of meiosis one, the centrioles begin to migrate towards opposite poles of the cell. All right. So they don't show this here, but it is happening. So we'll draw in some centrioles here. Remember what radiates out of the centrioles, or as this book refers to as them as the centrosomes, right? Microtubules, which will eventually create the apparatus or network of microtubules known as the mitotic spindle. However, this will not be a mitotic spindle, all right? This will be a meiotic spindle, if you will. These microtubules will be employed for the purpose of bringing about meiotic cell divisions. All right. So, as in pro-metaphase of mitosis, in prophase one of meiosis, the nuclear envelope begins to disintegrate in the same way. However, one very important distinction occurs. Okay, in prophase one of meiosis one, the pairs of homologous chromosomes interact with one another as they condense. Okay, so as this process of condensation of chromosomes into these bivalent X-shaped chromosomes occurs, the two homologous chromosomes for each pair of chromosomes, for each 23 different types of chromosomes, start to interact with one another in a very intimate way. This certainly does not occur in prophase or prometaphase of mitosis. All right. Recall that there are 23 pairs of homologous chromosomes, there are 22 pairs of autosomes, and 1 pair of sex chromosomes. For sex chromosomes, females have 2 X chromosomes, and for males, you have an X and a Y chromosome. So yes, it's true in that regard that X and Y chromosomes are not truly homologous, but one thing that I want to throw out to you right now is that however, at the tips of both the X and Y chromosomes, there are homologous regions between the two. And in that regard, we can consider the X and the Y chromosomes as homologous chromosomes, at least for certain pieces of genetic material at the ends or tips of their arms. All right. And just for the sake of classifications and bookkeeping and keeping things straight in our heads, we can go ahead and consider them, yes, to be another pair of homologous chromosomes. All right. Anyways, that's just thrown out there for you for some review as we go along. So in prophase 1 of meiosis 1, homologous chromosomes align with one another side by side. The homologous chromosomes become joined to one another by an elongated protein structure called the synaptonemal complex. Okay, so these two homologous chromosomes, which represent one of 23 different types of homologous chromosomes, including the X and Y chromosomes, they start to get very, very close to one another. All right, and they're joined together by this intricate protein complex called the synaptonemal complex. So as the homologous chromosomes join one another, they both continue to become increasingly condensed, much like how the bivalent chromosomes continue to become increasingly condensed all the way through prophase and prometaphase of mitosis. All right. The joined homologous chromosomes are together known as tetrads. All right. So all together, we have a tetrad. All right. These structures are known as tetrads because they are composed of a total of 4 sister chromatids. All right. So how many tetrads will form all together? Think about that for a few minutes. All right, so if each pair of homologous chromosomes of all the 23 different types of chromosomes get together and form these tetrads, all right, in which there's a total of 4 chromatids, how many total tetrads will we have? Right, we're going to have 23 because there are 23 pairs of homologous chromosomes. All right. So when the tetrads are fully formed, sister chromatids on different homologous chromosomes within the tetrad swap homologous segments of DNA with one another. I'm going to say that again. So when the tetrads are fully formed, sister chromatids, which happen to be on different homologous chromosomes within the tetrad, swap homologous segments of DNA with one another. Okay? These homologous segments of DNA contain the same genes in the same order. All right. However, keep in mind that these genes are not necessarily identical to one another. All right. Another thing I want to review when we're talking about homologous segments of DNA, um, what is the term that we use for the same types of genes that have slightly different sequences of nucleotides? This is going back way back when to the beginning. It's very important that you know this though. Alleles, right, alleles. So alleles are variations of the same type of gene. So when you're swapping homologous segments of DNA by the sister chromatids within the tetrads, different alleles are being swapped among the sister chromatids from the different homologous chromosomes. So here we have a depiction of what we just said right here. And you have to look really close. So remember, we said that the bivalent homologous chromosome of paternal origin is in blue, and then the bivalent chromosome of maternal origin is in white. All right, so now they cross together, they get really close, and then they swap the tips of their arms, okay? And remember, because all homologous chromosomes have the same types of genes in the same order, within these little tiny areas right here, and actually I'm going to switch to a finer tip on my pen here so we can outline this for you. All right. Well, it doesn't look like, you know what, that's right, you can't do this in PowerPoint. You can do it in Camtasia. You can select the point or the pen width, but you can't do it here. So let me just switch to a slightly different color. Let's do purple. So you can see right here that these tips will also, because they occur in the same areas of the respective homologous chromosomes, these tips will also contain the same types of genes in the same order. All right? Okay. So again, with the swapping of the homologous segments of DNA by the sister chromatids within the tetrads, different alleles, the same alleles, are swapped among sister chromatids from the different homologous chromosomes. All right? The process of genetic swapping between sister chromatids within the tetrads is known as crossing over or recombination. Okay? And you're going to have to be able to recognize these processes as both being the same thing. All right? The process is termed recombination as new combinations of different alleles are made on each sister chromatid. So I want you to look at your textbook for the rationale of crossing over recombination. How does changing the alleles within the sister chromatids better serve offspring? Well, what's the reason that we're doing this? You know, this is obviously some pretty complex cellular mechanics here. and it just didn't come out of the blue for no reason. There must be some type of driving evolutionary force that happened way back when, over 1 billion years ago, in all likelihood, that to be able to bring about this type of necessary complexity. Okay, so what do you think that driving force would be? Okay, how does this better serve the offspring, which will eventually inherit these different sister chromatids on these different homologous chromosomes? Okay, so I want you to continue to think about that question as we continue through the processes of meiosis 1 and meiosis 2. So the amount of crossing over between homologous chromosomes varies from chromosome to chromosome. For example, chromosome 1 will have about 2 to 3 crossing over events between each sister chromatid of the homologous chromosomes. As a result, each sister chromatid will have an average of 3 to 5 segments that alternate from maternal to paternal origin. So remember that one homologous chromosome is of maternal origin and that the other homologous chromosome is of paternal origin. And actually, let me draw a little diagram about exactly what I just said right there. So in order to illustrate this, I think I'm going to draw a new tetrad of my own over to the side. But first, what I'm going to do is I'm going to switch the colors over to colors that correspond with our designation of bivalent chromosomes of paternal versus maternal origin. Let me go pointer options. Ink color. Yeah, for some reason, there we go. My pointer disappeared. So we're going to go to ink color and let's go back to blue. All right. So let's draw in the bivalent chromosome from mom. Here's one of the sister chromatids here. And here's another sister chromatid right here, and they're held together by a, what is this structure called? A kinetochore, which holds together the two sister chromatids at their homologous, or identical rather, centromere regions of their DNA. All right, and now our other homologous chromosome is the one of paternal origin. Okay. Also a bivalent chromosome with two sister chromatids. Okay, and here's our kinetochore connecting the two together. Okay, but in this case we're going to color in these sister chromatids as being blue because these are of paternal origins. Ultimately came from this person's dad. All right. So if we were to look at the crossing over event, for example, of chromosome number 1, these two homologous chromosomes are going to kind of twist and turn, okay, upon themselves in such a way that each resulting chromatid on each homologous chromosome, okay, is going to alternate. I don't know why it's erasing this. Is going to alternate back and forth from paternal and maternal origin, okay? And then, so let's say the crossing over occurred between these two sister chromatids between different homologous chromosomes. Then this one would show the inverse pattern. So the tips would be dad. I don't know why this thing keeps on, it doesn't, switching back and forth between the mouse and the keypad on the computer. I think it's getting confused about what I want it to do. I just want it to draw. I don't want to do anything else. Sorry for this. So yeah, so right here I'm sorry, let me go back and erase that. That's not quite what I wanted to draw. All right, so this one right here is going to be the inverse, meaning it's going to have the opposite banding pattern than the other sister chromatid on the other homologous chromosome that it underwent crossing over with. So its tip would be from paternal origin as opposed to this one's tip is from maternal origin. All right. So I have no idea what's acting up about this. I'm still getting used to using this Wacom board. It's kind of annoying actually why every time I draw it creates this little halo around the picture. I don't want to zoom in. I don't want to do anything except for just draw this. So I don't know why it continuously asks me why I want to do this. All right, and so down here, because this end ends in a maternal tip, this end, this sister chromatid, which will be the inverse of this one, will end in a paternal tip. All right, so you can see where this right here, and actually let me switch the ink colors here. Pointer options, pen, ink color, red. All right. So what happened is a crossing over event occurred between these tips, this segment right here, these two segments 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 right here, and then these two segments right here, all right, to give us this kind of a banding pattern, alternating banding pattern of the sister chromatids. All right, and this is a little bit, or actually quite a bit simplified in reality because the crossing over is far, 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 far more likely to occur far out towards the tips of the arms, much more likely to occur in those locations that it is to occur near the centromere region. All right, and actually those likelihoods were the basic means by which geneticists were able to create initial and original maps of chromosomes and where different genes lied on different chromosomes. It has to do with these likelihoods of crossing over events from occurring. So again, just keep in mind that I made this look as if crossing over was equally likely in all these different locations of the sister chromatids of different homologous chromosomes when it is not. All right, and so the other sister chromatids, okay, on these different homologous chromosomes would also be crossing over with one another in a similar fashion. All right, in the same type of fashion where if they cross over at the tip here, okay, the tip from mom will end up on the tip from dad, and the tip from dad will end up on the tip from mom's sister chromatid, et cetera, et cetera. And and that same type of pattern, alternating pattern between maternal and paternal um, segments, homologous segments of DNA, would continue and be reciprocal between these two sister chromatids from opposite homologous chromosomes, which underwent crossing over events. All right, I know it's a little bit complex. Maybe if going back over this, it would help you a little bit. Um, but anyways, I just want you to appreciate is after this event, each of the sister chromatids on each of these homologous chromosomes look a lot different than they did when we first went into prophase 1. Okay, now they kind of have this zebra striped appearance, right? As a result of the swapping of different homologous sections of DNA among the sister chromatids of different homologous chromosomes. All right, this is the actual recombination. Okay, so we took the certain combination of alleles that we started off with in these sister chromatids of these homologous chromosomes, and we recombined them. Okay, we mixed up everything, did a whole bunch of gene rearranging, or I should say not gene rearranging, but better stated, allele rearranging, okay, between mom and dad's sister chromatids. Okay, we kind of scrambled things up, albeit in a very organized way, so that the alleles are still all in their same relative locations within the sister chromatids. It's just that you have different alleles um, in the sister chromatids now, okay, among this tetrad. All right? Okay. So after recombination is completed, the nuclear envelope dissolves and the recombined tetrads are pulled towards the metaphase plate by the same mechanism that lined up single chromosomes at the metaphase plate in mitosis, which, if you'll remember, is the mitotic spindle. All right, and we went over all the subtleties and complexities of the formation of the mitotic spindle and why, the basic biophysics of why the chromosomes line up at the middle. Except in this case, what's happening is that the tetrads are lining up in the middle as a result of the pulling and pushing of the mitotic spindle emanating from either centriole, or as the book says, centrosome at either pole of this cell. All right, and of course, it's important that the nuclear envelope dissolve, otherwise these microtubules of the mitotic spindle couldn't get at them. Okay, because remember, all these nuclear chromosomes, these bivalent chromosomes, are all in the nucleus. So we need to dissolve that nucleus so the mitotic spindle can reach out and grab them by their kinetochores and then also push past them and reach over to the other side of the cell and start this elaborate process of pushing and pulling that eventually lines up these tetrads, these 23 different tetrads, right in the middle, okay, at the metaphase plate or equatorial plane, if you will, of this cell. All right. Again, it's the same thing that's happening as we talked about before in mitosis, except that instead of individual bivalent chromosomes all arranging in a line here, now instead we have 23 homologous pairs of chromosomes joined together to form tetrads lining up here at the metaphase plate. And I'm just going to change the ink color to red as we go along. All right. So the phase in which the 23 tetrads are aligned with a metaphase plate or equatorial plate, as we said before, is known as metaphase 1 of meiosis 1. As you can probably guess where this is headed, we do this only so that we can break these structures that we have formed apart, okay? But whereas in mitosis, we are breaking apart the actual bivalent chromosomes, separating those sister chromatids so that each one becomes a, do you remember right, so each sister chromatid magically becomes called a daughter chromosome. That isn't happening here. We aren't splitting apart sister chromatids here. Instead, we're breaking apart that synaptonymal complex that formed. Remember that synaptonymal complex that formed. Oh, that's a really bad picture, I'm sorry. That synaptonymal complex that formed between the two homologous chromosomes, we're breaking that apart, okay, so that the two fully intact bivalent chromosomes, okay, each homologous chromosome for a single type of chromosome goes flying in opposite directions towards the opposite poles, okay, of the cell, which are designated by these centrioles, okay, which the mitotic spindle, or not the mitotic spindle, but the spindle apparatus is emanating out of. You see why I guarded myself? I didn't want to say mitotic spindle because this is not mitosis, this is meiosis. All right, and as soon as that happens, we go into telophase 1 of meiosis 1. And telophase 1 of meiosis 1 is basically the same type of deal going on in telophase of mitosis. The first of all, as the chromosomes collect in the different locations, they are the different poles of the cells. Rather, they begun to decondense, and then two new nuclei will form around them. Okay. This, however, contains quite a few differences that we need to go over, and some different terms thrown in there as well. So let's go back and look at anaphase 1. All right, I'm sorry to go back and forth a little bit like this. This diagram has had quite a bit of brevity built into it by the publisher. I don't know why, just to conserve space, or they assume that that people can fill in the blanks on their own. But if you notice here, it does not contain telophase as part of the diagram, but telophase is definitely in there. All right. So let's talk about anaphase in just a little bit more detail before we go into telophase, though. So as the mitotic spindle applies increasing and equal tension upon the different homologous chromosomes, within each tetrad, the homologous chromosomes pull away from one another. The phase at which these recombined homologous chromosomes are pulled away from each other is also known as anaphase 1 of meiosis 1. The process, however, the process of the actual pulling apart of the homologous chromosomes and breaking them away, or breaking apart the synaptonymal complex, okay, is known as disjunction. All right, disjunction. So the process by which these homologous chromosomes are broken apart from one another is known as disjunction. All right. The process of disjunction is important, as mistakes here made can be devastating. In rare cases, both homologous chromosomes within a single tetrad can fail to separate. Okay, so if they fail to separate, an entire tetrad may go to one pole of the cell, or it may be pulled to the other pole of the cell. And that's bad, because one cell, or one pole, which will become a new cell later on, as you can see here, as a result of telophase, all right, will be placed within one of these cells, unless or missing genetic information will be the result in the opposite cell. So again, as a result of errors in disjunction, and errors of having these two homologous chromosomes separate, you're going to get extra genetic material in one of the resulting cells and less genetic material in the other resulting cell. All right. Does that make sense to everybody? Okay, the failure of the two homologous chromosomes within a tetrad to separate is termed missegregation. Missegregation results in the erroneous process of a non-disjunction. Non-disjunction, okay? So if we have, or rather, let me say this, let me put a big X to symbolize that these two never properly separated from one another, okay? And so the whole thing gets pulled over to one pole of the cell during anaphase, all right? So here we have anaphase, and these guys are all still connected together within a tetrad, and that whole tetrad gets yanked to one pole of the cell, all right? That whole entire tetrad is going to end up in only one of the cells, okay? Instead of being evenly dispersed, each bivalent chromosome in the tetrad being evenly dispersed from the two resulting cells, an entire tetrad worth of chromosomal material is going to be in just one of the cells. All right. So this problem or this failure is known as missegregation. All right. Missegregation results in the erroneous process of non-disjunction, okay? So when you do not have even dispersal or equal dispersal of the two homologous chromosomes within a tetrad to either of the resulting cells of meiosis 1, we call that missegregation, okay? Missegregation is one specific error, okay, within a larger process termed non-disjunction. So missegregation, okay, meaning a failure for the chromosomes within the tetrad to split or segregate, so the failure of the chromosomes within the tetrad to separate or segregate results in an erroneous process known as non-disjunction. Okay. The erroneous process of non-disjunction. All right. And as we said before, non-disjunction can result in chromosomal disorders in the offspring resulting from a gamete, okay, or a sperm and egg. All right, non-disjunction can result in chromosomal disorders in the offspring resulting from a gamete that was malformed in this process. Non-disjunction can result in chromosomal disorders in the offspring who resulted from a gamete that was subjected to this malformation. Okay, so if the problem, okay, of having extra genetic material in this cell, which will eventually go on to become a certain gamete, okay, or the problem of having less genetic information in this cell, which will also go on eventually to become another gamete, or may or may not go on to become a gamete in the case of a female, then whatever zygote someday is the result of a gamete that was formed through this erroneous process, all right, will be at serious risk of having increased or decreased genetic material on the level of an entire chromosome or bits and pieces of a chromosome. All right, and that can result in devastating genetic problems, okay, that fall under the category of chromosomal disorders. And some of you are going to be doing chromosomal disorders for your research papers, and if you're not doing chromosomal disorders for your research papers, you will be watching presentations from your peers who have done research in such areas. Um, and also we will be going over the specifics, the mechanics of non-disjunction errors that bring about some of these disorders as we go on in class. Alrighty. So after anaphase 1 of meiosis 1, telophase 1 of meiosis 1 begins, as we already said, as you probably already intuited before I even said it though. Just as in mitosis, telophase 1 of meiosis 1 involves the reformation of the daughter nuclei, as we said before, all right, and decondensation of the genetic material back into chromatin. As you can see, by the time we end telophase here, both of these daughter nuclei have their DNA reformed back in the decondensed form of chromatin. All right. However, another very significant difference exists in telophase of meiosis 1. In mitosis, individual daughter chromatids for each of the different 46 chromosomes decondense in each daughter nucleus. Therefore, each daughter nucleus after telophase of mitosis has a diploid set of chromosomes. There's 46 total chromosomes. All right, there's 46 chromosomes in each daughter cell at the end of mitosis. But in telophase 1 of meiosis 1, only 23 individual chromosomes, which represent one from each of the 23 types, decondense into chromatin. So why is this? Well, if you recall, in anaphase 1 of meiosis 1, each individual homologous chromosome in each of the 23 tetrads migrates to the opposite pole of the cell. All right, we do not have chromosomes being split in half into, you don't have their daughter chromatids being broken in half and then those daughter chromatids becoming their own daughter chromosomes. Instead, the whole homologous chromosome from maternal origin and the whole homologous chromosome from paternal origin, albeit they're all kind of recombined at this point, split apart from one another. All right, and we said how many total pairs of homologous chromosomes do we have? 23. So how many tetrads do we have? 23. So when we split all the tetrads apart, how many chromosomes are we going to have in either of these daughter cells that result? 23. All right, and because of the homologous pairs, only one of the homologous pairs goes to this cell and only one of the homologous pairs goes to that cell. Do we have homologous pairs in the resulting daughter cells that result from meiosis 1? No, we don't have the homologous chromosomes anymore. So we've effectively cut the genome into half. We've gone from a diploid genome, okay, to a haploid genome in the two resulting daughter cells of meiosis 1. Oh, I don't know why that happened, but I'm glad it did. Okay. So, oh, I see. I moved to the next slide, so it got rid of all of my junk on the other slide. Yeah, there, it all returns again. But we will get rid of it all shortly. Okay. So again, recall that the two new cells resulting from mitosis are diploid, as we already said. The two daughter cells resulting from mitosis are diploid. However, the cells resulting from the first division of meiosis, which is called meiosis 1, are haploid, as we just demonstrated. So in other words, there are half the number of chromosomes in each cell after the first division of meiosis, okay, compared to um, uh, mitosis, okay, where the total number of chromosomes is retained after the division of the cells. So therefore, meiosis 1, because you cut the number of chromosomes in half and you go from the diploid state to the haploid state, all right, within meiosis 1 is often referred to as the reduction division. Let's see if I can draw this sideways here. Oh, wow, pretty. The reduction division. No, that's not right. There's an I and some extra letters in there. Anyways, well, I almost wrote it sideways. That's pretty good. You can see why I have to write in a lot of things ahead of time, because if I used this just solely like it was a chalkboard, it would be pretty difficult for everybody out there to understand. So anyways, that's why we call meiosis 1 the reduction division, okay? So after telophase 1 of meiosis 1, the two resulting daughter cells, which we have here, transition to interphase, okay? Interphase of still meiosis 1, all right? And by the way, in the sperm, these cells, in the male rather, not in the sperm. We haven't made sperm yet. In the male, these two resulting daughter cells of meiosis 1, all right. Ooh, we're zooming in. I don't know how I did that, but, oh, I see. So if I still have to go over how we zoom in. That's going to be clever and useful for later on when things get more complex, though. But anyways, so in the male, these cells are called secondary spermatocytes, okay? So these two cells right here are called secondary spermatocytes in the male, okay? And in the male, both of these secondary spermatocytes will proceed to meiosis 2, okay? Both of these secondary spermatocytes, which are the daughter cells that resulted from meiosis 1, will progress to mitosis 2. All right. In the females, however, and let me go erase a lot of this junk so we can better write in what's going on in the females. In the female gonads, aka the ovaries, the two daughter cells that interphase, or that enter interphase of meiosis 1 have two different fates. When the first meiotic division occurs, there is asymmetric distribution of cytoplasm. So as a result, one of the cells resulting from meiosis 1 in the female is very small, while the other is quite large. So you have asymmetric distribution of cytoplasm here in the female during the meiosis 1 division. All right. So one of these cells, and actually let me change the color here to blue. So one of these cells is going to be quite large, okay, where the other cell is going to be quite small, okay? Both of these cells are going to contain the same type of genetic material. It's just that one of the cells inherited the vast majority of the cytoplasm. Okay, this one cell that inherited most of the cytoplasm is going to be referred to as the secondary oocyte, and it is analogous to the two secondary spermatocytes that are formed after the first meiotic division in the male gonads. All right. Okay. The secondary oocyte will proceed to meiosis II, just like the two secondary spermatocytes will proceed to meiosis II. All right, but in this case, you only have one of the cells, the secondary oocyte, the one secondary oocyte in the female that is able to proceed to meiosis II. And eventually that cell will be the one that gives rise to the egg. The other smaller cell in the female resulting from meiosis I is referred to as the first polar body. Okay, the first polar body. It will continue to reside alongside the secondary oocyte. It's going to go along for the ride, but it will never be fertilized by sperm, like eventually the progeny of this secondary oocyte will. Okay, it will never result in the gamete, okay, that will participate in fertilization. It's just going to forever and always, until it just atrophies and dies, be called a first polar body. Okay, the first polar body will not enter meiosis II. So interphase 1 of meiosis I proceeds very differently from interphase of mitosis. There is no S phase in interphase of meiosis I. There is no S phase in interphase of meiosis I. There is no S phase in interphase of meiosis. I stuck another I in there that's not supposed to be there. There is no S phase in interphase of meiosis I. All right. The DNA of each chromosome is not replicated. This is a very important fact to remember. In interphase 1 of meiosis I, the chromosome number is now half of what it was in meiosis I, or when meiosis I was entered. So in interphase 1 of meiosis, or rather in interphase of meiosis I, all right, in interphase of meiosis I, the chromosome number is now half of what it was when we originally entered meiosis I, right? And it's because of that, because right here where we transitioned from light blue to purple, we underwent what is commonly referred to as the reduction division, all right? We reduced the number of chromosomes in half. We went from the diploid state to the haploid state, okay? So, while we did cut the number of chromosomes in half in each of these daughter cells, each chromosome has still two sister chromatids within each chromosome. Okay, so each chromosome still has two sister chromatids, is a better way to say it. Each chromosome still has two sister chromatids, right? Why? Because look, this guy right here went here, and then this guy here went here. I don't see any separation of the sister chromatids. That never happened like it does in mitosis. Never happens. Kinetic core is still intact. Sister chromatids still intact. Bivalent chromosomes still intact. And so they go in here, all right, so that you still have two identical copies of each of the 23 different types of chromosomes. It's just that you no longer have homologous chromosomes for each type of chromosome. Okay, so there's no point in doing S phase because you already have identical copies. Now, here's a very, 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 very important thing to keep in mind, what I just said. And it does contradict what I just said to some degree. All right, but some things we say for purposes of just trying to outline a general working logic. All right, a place to begin with. So now I'm going to throw something out there that might be kind of a little bit of a ratchet in the machinery that's developing. But keep in mind that because of recombination, the sister chromatids are not completely identical. All right, each two sister chromatid within the 23 chromosomes, the 23 different types of chromosomes that each of these daughter cells have, are not 100% identical copies of one another because of all of this crossing over that occurred in prophase 1 of meiosis 1. Okay, it's as a result of the recombination that occurred in prophase 1 of meiosis 1 that no sister chromatid within either of these daughter cells is completely 100% exactly alike. All right. So in the next slide, we'll tackle meiosis 2, the second meiotic division. We are almost there. So hopefully all of this stuff just disappears when I go to the next slide. Yes, good. All right. The cell division of meiosis 2 is similar to the process that occurs in mitosis. Okay. However, unlike somatic cells undergoing mitosis, the secondary spermatocytes and secondary oocyte that undergoes meiosis 2 only have haploid sets of chromosomes. So why do they only have haploid sets of chromosomes? Why is that the two secondary spermatocytes in the male and the one secondary oocyte in the female only have haploid sets of chromosomes? Well, we just went over it. Right, because the diploid set of chromosomes was cut in half to a haploid set of chromosomes during meiosis 1. Again, recall that meiosis 1 is often referred to the reduction division for this reason. Okay? In contrast to mitosis, the two sister chromatids within each of the 23 chromosomes are not 100% identical to one another. Okay? Again, we just said this before, but I'm repeating it now, I believe, for the fourth time because it's just a very important concept that I want you to really internalize. All right? Believe me, it's going to make things a lot easier as we give reference to these events later on in the class. Okay, so in contrast to mitosis, the two sister chromatids within each of the 23 chromosomes, within each of the 23 bivalent chromosomes, okay, are not 100% identical to one another. The sister chromatids within each chromosome are not 100% identical to one another because of crossing over or recombination that occurred in prophase 1 of meiosis 1. Okay? So as you can see, we enter meiosis 2 just as we left off, in interphase of meiosis 1. Okay? So recall that this diagram showing two cells in interphase of meiosis 1, all right, here's 1 and 2, would be most accurate in the setting of meiosis within the male gonads or testes. Why is this? Because recall in meiosis 1, the male produces two secondary spermatocytes. These cells in interphase right here, where's my little marker? These cells in interphase, of which there are two, best reflect the situation that you would have in the males because you have two secondary spermatocytes, all right, resulting from the first meiotic division. However, recall that in the female gonads or the ovaries, meiosis 1 results in one secondary oocyte and what else? One polar body, okay? Also recall that the secondary oocyte only proceeds to the meiosis II division. Okay, so therefore, if we were going to use this figure right here to demonstrate the second meiotic division, or meiosis II, in the female gonads, we need to keep in mind that only one of these two cells, the ones labeled as an interphase, will actually undergo meiosis II. In the female gonads, the one cell resulting from meiosis I that will undergo meiosis II is the secondary oocyte. All right? The other cell resulting from meiosis I is the first polar body. The first polar body will not undergo meiosis II. It will always and forever be a first polar body, and then it will just atrophy and die eventually. Okay, it will never go on to become a gamete. It'll never amount to anything. Okay, so from interphase, the two secondary spermatocytes in males, and by the way, this big arrow is just to help you make the connection that what's going on down here is exactly what we find ourselves in up here in this gray region, okay, which commences the beginning of meiosis II now, okay. From interphase, the two secondary spermatocytes in males and one secondary oocyte in females progress through prophase 2, all right, metaphase 2, and anaphase 2, okay, and telophase 2, which is not really explicitly shown, but it's there as we transition from anaphase 2 to the actual gametes, which is this is all leading up to. This progression from prophase 2, metaphase 2, anaphase 2, and telophase 2 in the two secondary spermatocytes and one secondary oocyte is much in the same vein as the processes that were conducted in mitosis. So for a review of these mechanisms, okay, of taking the, or going through chromosome condensation, reforming the bivalent X-shaped chromosomes, disintegrating the nucleus, forming the spindle complex, all right, having the chromosomes, the 23 different bivalent chromosomes line up at the metaphase plate, all right, and only 23 here where you would have 46 of them lining up in the case of mitosis. But here, because of the reduction division in meiosis I, we only have 23 chromosomes lining up, the 23 different chromosomes, that is, lining up at the metaphase plates, okay, in these two situations. And then the way that the mitotic spindle separates the sister chromatids in each bivalent chromosome so that they each become daughter chromosomes from here on end and get pulled apart to opposite parts of the cell. The mechanics are all exactly the same as mitosis. All right? So for a review of the mechanisms that conduct these phases in mitosis, please revisit the mitosis component of this lecture. All right, which actually would be segment 2 of the chapter 2, part 3 lecture series. When telophase 2 of meiosis II is complete in the male, 4 gametes, all right, are going to be the result, okay? 2 from each secondary oocyte, or what was a secondary, or not, I'm sorry, from secondary spermatocytes, okay? So each of these guys right up here were secondary spermatocytes once upon a time, right? All right, and so eventually both of these secondary spermatocytes resulted in the formation of 2 gametes as a result of meiosis II, okay, giving us a total of 2 times 2 equals 4. 4 gametes. All right. Okay. Within each gamete, or what is known as a spermatid, kind of an undeveloped sperm, within each of these spermatids, um, there is a haploid set of single daughter chromosomes. There is no more bivalent chromosomes with sister chromatids because we split them in anaphase 2 of meiosis 2. See this? So we used to have bivalent chromosomes here with two sister chromatids. They're not actually identical anymore because of recombination that occurred in meiosis 1, but they are pretty somewhat close anyways, depending on the chromosome that we're dealing with and how many crossing over events occurred. But when they separate to become daughter chromosomes, okay, it results in each of the resulting gametes of having only a singular copy, okay, of each chromosome within their haploid set of chromosomes. Does that make sense to everybody? You might need to put the video on pause and go back and go over what I just said if it's still confusing to you, and maybe go back and do it a couple times. And if it's still a problem, then please come and see me. Again, this stuff is not easy. I realize it's not easy. It's very confusing. There's a lot of new jargon interjecting with it as well, and that's why I go so slowly with this material at first, because I really, really, really want you to try to get a good handle on it, because it's going to make your life so much easier as we progress. Okay, so the gametes or spermatids then go on to mature within the seminiferous tubules of the testes, and hopefully you've already learned about them either in general biology or in anatomy and physiology. If you've taken anatomy and physiology yet, from here on end, they're released into the seminiferous tubules. And you're going to have to go back to anatomy and physiology to review how they eventually get out of the male body from there. All right, that's where this class leaves off, and A&P takes the baton. All right, so when telophase 2 of meiosis 2 is complete in the female, recall that only one, okay, secondary oocyte is actually going to participate in meiosis 2, okay? So, what's going to happen here is after this one secondary oocyte undergoes meiosis 2, you're eventually only going to get one solitary ovum or egg. And the split is actually going to be asymmetric in its distribution of cytoplasm, much as it was in the female in meiosis 1. So what I'm going to do is I'm going to try to get the eraser here and get rid of this, because I didn't mean to make this female gamete, or what I will show as a female gamete, as being so small. All right, so the only one female gamete that results, okay, from meiosis 2 is going to be quite sizable, all right, compared to the other cell that results. Okay, and what is this second cell that results? What is it called? All right, well, I'm glad you asked. Well, this guy right here is going to be the egg. This other cell resulting from the meiosis 2 division is called the second polar body, okay? And in females, none of this even occurs, right? Because the first polar body just kind of, is completely outside of the loop at this point. It's there, but it's not really, it never participates in meiosis 2. Okay, so in the female, we have this final division from meiosis 2 results in a large egg and a tiny secondary polar body. The egg is the actual gamete. the secondary polar body is, oh, I put bod. Polar body is just like the first polar body in that it is a non-participant. Okay, it exists, but it never actually gets to participate in fertilization. Okay, so the solitary ovum or egg, like the spermatid, possesses a haploid set of single daughter chromosomes. Each daughter chromosome is the direct descendant of one of the former non-identical sister chromatids within the chromosomes of the secondary oocytes. Okay, just one of the chromatids within what used to be the secondary oocyte. Okay. The process of fertilization involves the combination of the sperm and the egg. When this occurs, two haploid chromosome sets of the sperm and the egg combine to form a completely new and unique diploid set of chromosomes and a totally new cell. The resulting cell is called a zygote. So from here, the zygote will undergo successive cycles of mitosis to yield a developing multicellular embryo. The embryo cells will continue to divide and divide and divide via mitosis and give way to the developing and maturing fetus. So speaking of mitosis, this would be a great opportunity to go back and review mitosis. After that, you can review meiosis, and then mitosis, and then meiosis again. All right, so eventually you guys are going to be able to draw this all out without even thinking about it. Well, at least be able to recognize it when other people give reference to it without having to do too much recollection or it causing too much confusion. All right, that's the end goal of this. So it's going to take a couple more cycles of memorization or attempts at memorization before it really becomes internalized. So again, hopefully watching these videos over and over and over again, or maybe even running them through your iPod, okay, so that you can be listening to me droning on and on and on about this stuff while you're trying to accomplish some other task. Maybe even that could have some benefit to it. At least that would create questions that you could then go back and answer when you have more time to dedicate exclusively to this. So that's where I'm going to end you. Hopefully we haven't gone too far over our time limit. And I will be meeting you again for our discussion, the first of many actually, on the topic of gene expression. So I will see you then.